Okay, next subtopic in polymerase chain reaction is its advantages, disadvantages, and its applications. So first of all, advantages and disadvantages. This PCR is very quick. We have earlier discussed that this is possible in a matter of hours. So we can quantify, we can amplify different genes in a matter of hours. This is reliable. We know that, okay, the gene expressed is the desired one. It's not some other gene. So it is reliable. It is sensitive. If we have only 1 or 2 copies of that particular gene, even then we are able to amplify that particular gene. It's relatively easy. Only few reagents are needed. Only one instrument is needed. We need only one source of heat. That's all. So it's relatively easy. It's not complicated. Its procedure is not so difficult, and it is specific. It means that we can amplify the desired gene, and in this process, some other gene that is not expressed. It is specific. Disadvantages. It is expensive at the same time. When we say it is expensive, it means it involves expensive instrument, which is PCR machine or thermocycling machine. It has the price of about 1 million, so 1 million rupees. So we can say it is expensive according to our economic situation. There may be chances of contamination. So some other bacteria may grow, and these bacteria may lead to false positive reactions. So if there is contamination. Similarly, there may be false reactions, like we discussed. If there is some contamination, we may get expression of some other gene, of course, which may lead to wrong results. Sometimes there is problem that unspecific gene that is also amplified. So although it is specific, but sometimes due to some problems, other genes may also be expressed, may also be amplified. So we should keep these things in mind. Applications, how and where we used this PCR process. We use PCR process in testing and in diagnosis of various diseases. For example, in thalassemia or other diseases, we are able to diagnose the disease at early stage to prevent it at later stage. So different other diseases, they may also be diagnosed with the help of this technique. DNA may be cloned. The desired gene which we want to clone, which we want to express, that may be cloned by this technique. This technique may also be used in gene expression studies. We want to see whether this particular gene is expressed or not, and if it is expressed, what type of changes this gene may be associated with. So we can also use this technique in studying gene expression. Similarly, this technique may be used in amplification of rare DNA, like that of fossils. So if we want to study animals and plants of remote past time, for example dinosaurs, so then we are able to amplify the DNA of those organisms, or some part of the DNA of those organisms. So rare DNA of the remote past animals or plants may be studied with the help of this technique. This technique may also be used in forensic DNA analysis. For example, at crime scenes, we are able to see that this particular hair or this particular part of the body or this spot of the blood belongs to which person. So with the use of this technique, we can identify that this blood spot or this hair belongs to which person. We can use this technique in case of paternity testing. So in case of disputed paternity cases, this technique may be used. And similarly, we can also use this technique in identification of cause of various diseases in human beings.